Hello, everybody. So new topic, 3.5, genetics, modification and biotechnology. This topic, it's easy to break it down into three groups. At least for me it is. So the first five, if we look at these, so these first five assessment statements are kind of all talking about the same thing. So they're talking about how we can take a little bit of DNA and we can use it to identify things. So we can, figure out someone's genetic code or who a drop of blood belongs to. We can use it for paternity cases. So all of this talks about that. The second part is all about genetic modification. So that's up here. So genetic modification is taking a gene from one organism and putting it into another organism. So we do this with our foods for sure, and also with some animals. And then the last little section is about clones. So where clones exist in nature, where clones, like how do we make clones and how they're used. So that's that last little piece here. And then there's some little experiment that we need to do at the end. So that's 3.5. In this first part of 3.5, I'm only going to talk about one slide. Let me get there. And it's called polymerase chain reaction. So in this assessment statement, it's asking us to outline the process. So it's actually quite simple, and it's very similar to DNA replication. Basically, it is DNA replication, except we're controlling it. I think the assessment question is a little bit, or the assessment statement is a little bit confusing. So it says outline the use of polymerase chain reaction, and you can just use the abbreviation PCR to copy and amplify minute, so that's small, so small quantities of DNA. So as I said before, we would do this if we found a drop of blood at a crime scene and we wanted to know who it belonged to, like the murderer or something. And the process is real simple. So I don't know if you can see here, but the temperature is 95 degrees. So what they do is they take DNA, they take our sample of DNA, and that's what this is. It was once joined together in this picture. And they heat it, and that heat will break the hydrogen bonds. If you can see this is like one backbone. So if you remember, it's like phosphate to the sugar to the nucleotide. So this nucleotide, this rectangle here, are all these yellow pieces. So these are all our A's, T's, C's, and G's. And when we heat it, we break the hydrogen bonds, and our DNA separates. Then what we do is we add a primer to one end of it. So that's this piece right here. We add a primer to one end of it to identify the region that we want to copy, because we don't want to copy all the DNA. We just want to copy a little piece of it, because even that little piece will be unique to that individual or that organism that we're looking at. And then all these nucleotides, they're floating around, and they're going to form complementary base pairs to their exposed partner, their exposed complement. So if we had an A here, a T would join. And this just, it binds to it. But if you remember from DNA replication, do you remember when we were talking about like how we can add our nucleotides, so phosphate, sugar, and then the nitrogenous base, let's pretend that's adenine. And then another one binds here, phosphate, sugar, and another nitrogenous base. We need an enzyme here to connect this phosphate to the sugar above it. And to do that, we need DNA polymerase. That's its job, is to join these. In PCR, the difference would be we use something called Taq polymerase. Taq stands for like thermal aqueous something. So it's an enzyme that bacteria use at thermal vents. And because they live at thermal vents, they can withstand such high temperatures. So this Taq polymerase will join all those bases together, and after one trial, you'll get 2 new pieces of DNA, and you just do it over and over until you have all the DNA you need. That's PCR.